Okay, welcome everybody. Nice to see smiling faces and masks, and people are working, and that's great. My name is Janet Boardwin. I'm the career director at the College of Natural Sciences at UMass Amherst. We are thrilled to have representatives from Seres Therapeutics here to tell us about their organization, who they're hiring, what the practices are. And we have many scientists and representatives from there. If you are from Seres, you know you are signing on as Julia Williams. So feel free to change your name in the right chat of your box. Any UMass Amherst students, if you're able to keep your cameras on, that would be great. It's always helpful to presenters to see faces and expressions. Even if you're working and have masks, that's fine. If you're unable to, we absolutely understand that. It's just helpful to the presenters. So I'm now going to turn it over to our Seres contingent. Thank you again for coming and your interest in recruiting our students at UMass Amherst.
Super, thanks for that introduction, Janet. So I think the first thing that would be helpful for folks, we'll just briefly introduce all the Seres folks that are on the call today. My name's Chris McAllicher. I'm the Executive Director of Technical Development at Seres. I cover analytical development, bioprocessing, and formulations. And I've been with the company for over 7 years. Lisa, would you like to introduce yourself?
Sure. My name is Lisa Lonis. I'm the Executive Director of Talent Acquisition here at Seres Therapeutics. I joined Seres in November, so I am a brand new employee, and it's been really interesting assimilating into an organization virtually. But I'm really excited to be here, and I'll talk a little bit about why I made the decision to join Seres as we move through the presentation.
I see Andrew has signed on.
Yeah, hi, my name is Andrew Billings. I'm a Principal Research Associate in Bioprocessing. And I've been at the company for 4.5 years, and I'm a UMass Amherst alum.
Thanks, Andrew. I see Ariel's here.
Hello, my name is Ariel Ryder. I've been at Seres now for about 6.5 years. I'm a supervisor in the analytical operations group, which was within the analytical development group. Yeah, and I think I'll pass it off to, I don't know who's next.
The real Julia, perhaps? I think we have the real Julia online.
Hi. Hello, everyone. I'm the real Julia Williams. I'm the admin for the CMC department. And so I'm sort of a jack of all trades. I'll help out with recruiting. I help out with interview scheduling. If you have any questions, I'll be here in the chat every now and then. If people have any questions, I'll probably just jot them down so everyone can see it. If you have questions, if you do have any questions, just ping me directly, and I can also just post them in the chat. If you have any questions, like maybe you don't want to voice it, I know sometimes speaking, on Zoom is a little bit stressful. So I'm here to help you. And Melvin.
My name is Melvin. I'm the recruiting coordinator here. So I do a lot of the scheduling for interviews and communicating with candidates as they move through the process. So, if you're interested about any of the careers on our website, I'm the person you can go to and send a resume to, and I'll get you in the right direction. I joined Seres in October, and it's been a lot of fun joining an organization, growing the way we are, and being pioneers in the industry as well. So definitely feel free to ask me about questions regarding that as well. Happy to do so.
Thanks, Melvin. I think the last one on the line is Melissa. Did I see you hop on?
Yes, I was Julia, but now I'm Melissa again. So I'm Melissa G, and I lead the team for donor analytical sciences, and that's basically screening donors for potential microbiomes of interest for us. And I have a couple of positions open. And I have been at Seres for just since November, or really the end of November, effectively December. And I ditto the responses that it's been very interesting coming on board virtually, but a very extremely exciting time to be building this company right now, growing and all the good work that we need to do in order to get product out to patients. And I think that that'll become clear as I think Chris will be speaking about shortly. So.
Fantastic. Thanks, folks. So yeah, please feel free to connect with any of us after after this session. We'll have some question and answer time at the end. What we've got scheduled for today is a kind of a short presentation, introduction to the field of microbiome therapies. We're an interesting and emerging industry, and we think we're pioneers in the field. And so we'll give you some company background as well, and then Lisa will also speak to some aspects of our corporate culture, company culture, benefits and packages, those kinds of things, and then also how we're responding to life in this time of coronavirus, which is another thing altogether. So we'll get going. All right, are we on? Which one are we on?
You're on the yeah, I think you're on the cover slide now, Chris.
Is it showing presenter view or?
Yeah, it's presenter.
All right, sorry, there we go. Had it right the first time. All right, so we're gonna introduce Seres. We're gonna introduce, most of us are in Seres CMC. And so, that's a very specific department and group within Seres Pharmaceuticals, so Seres Therapeutics, and we'll go through that today. And very excited to get your questions at any time throughout the presentation. Go ahead and either speak up or prod Julia in the chat that you'd like to interrupt my speaking. So we're looking at a very specific function within the body, and that's all the things that bacteria do that exist within the larger context of all the other functions of the body. And what we've found out through the Human Microbiome Project is that there perhaps as many or more bacterial cells in and around the body, either inside the gut, on the surface, skin, ears, nose, throat, than there are actually human cells on the body, right? And I think this has been a fairly well established and communicated aspect in popular science and popular culture. And it's really what motivated our company, Seres Therapeutics, to start as a small fledgling startup company about 9 years ago. What we're looking to do is understand how the population dynamics of those bacteria provide resistance to pathogens, impact our body's response to inflammation or our immune systems, and how they could regulate certain aspects of our metabolism, either directly through the way that we handle nutrition, or indirectly through small molecules that may impact the way our body functions in other ways. And so we know that the microbiome is implicated directly in human disease. We're looking at C. diff infections. This is a hospital-acquired infection, probably the top or amongst the top of the nosocomial infections. And we've been able to clearly associate that when a gut bacterial system is what we call dysbiotic, it becomes susceptible to a C. diff infection. We're trying to also connect the microbiome to inflammatory diseases, the way body can respond to cancer, and so we've got active research in all of these areas. Our approach to drugging the microbiome, right, and that is using some sort of therapy to alter the way a patient's microbiome is acting, is through the incorporation and introduction of new microbial species that perhaps a patient is deficient in. And so what we are is a classic microbiology shop at our core. We've got tens of thousands of isolates, hundreds and hundreds of strains, from all sorts of healthy people, fresh isolates. They're not adulterated from from extensive domestication, laboratory domestication, or direct genetic manipulation. We're not dealing with genetically modified organisms. Those are perfectly valid ways of perhaps approaching the problem, but we're thinking about it more as a microbiological problem. And we're trying to capture in this description of a consortia, what is the natural breadth and diversity of a consortia, and how do we complement it in people who are deficient in some way? And then we're aiming to make drugs formulated for oral delivery using the elevated standards of the good manufacturing practices outlined by the FDA or other regulatory bodies around the world. As we think about our drugs, the easiest way to think about it in a lot of ways is the infectious disease area. And here we have a nice sequence cartoon where we show the C. diff bugs perhaps, or other bad actor species are here in orange. What we're trying to do is deliver a series cocktail of bacterial consortia, which can bring life back to a healthy and diverse ecosystem. Those bugs are going to enter orally into the alimentary canal and eventually end up in the colon, where we're trying to do most of our work. Eventually, those species will engraft, which means really take hold and start replicating, and then crowd out and compete bad actor bugs. And then eventually, after we've got those compositions restored, we start seeing those secondary metabolic effects, and perhaps those are bile acids or short chain fatty acids. We're very interested in tryptophan metabolites and indoles, and then maybe some other distant effects and protein interactions. And so that's the easiest way to conceptualize. In reality, identifying bad actor bugs is not as simple as it sounds. In the cases where it's pathogenic, 10% of the population perhaps is carrying C. diff at any time as a non-pathogenic participant. And so, there's some other aspects here where really it's a combination of the presence of pathogens with this dysbiotic character, or it's perhaps even more complicated. There might not be a singular bad actor species, but just an overabundance of certain kinds of activity which are causing inflammation or stress in the GI tract. And so the infectious disease component is easy to think about it, but we're thinking about it also in those complex states around inflammation and interaction with the immune system. This is the field has gotten very complicated. This is a pictogram kind of circa 2018 of all the different companies that have emerged in the field. And I mentioned there are lots of ways to drug the microbiome. We're up here at the top. We deal in two specific kinds of live biotherapeutics. We use biologically sourced material, purified healthy donor samples, as well as fermented systems where we're using discrete mixtures of well-characterized and cultivated bacteria. Other companies are using prebiotics, trying to influence the development of a microbiome shift through food, right? Through food and fiber. Other companies are looking at single strains, which might provide a silver bullet to some issue. Other companies are working on engineered bacteria, specifically where you've got to overexpress a pathway. So companies might be looking at PKU or oxalic acid or some other overabundant chemical compound which is causing an issue in patients, and then trying to prevent that issue from taking hold because of by overexpressing some consumption pathway. Other places are looking at very exotic phage systems, which are like bacteria viruses, or other antibiotics which are meant to target certain aspects of the system. But so we're here up top. We put ourselves top and center in the diagram, but really focus on those live biotherapeutics, bacteria, all microbiology, no genetically modified organisms, and then complex mixtures, because that's how we think we're going to solve the problems of a wide array of patients. There's a lot that needs to be developed in this field. Saying that you're going to put a bunch of bacteria in a capsule and give it to people is kind of the simple way to say it. We've spent a lot of time building an internal system, which is vertically integrated. As a field leader, we found that people were unprepared to support us. A lot of times you're running some tests or early manufacturing is happening outside of your organization at a contract research facility or a contract manufacturer. What we've done is build everything internally, whether it comes from clinical samples and working with academic health centers to get patient samples as we explore new diseases, whether it's developing our own ex vivo or in vivo disease models, our own computational bioinformatics and bioinformatics, and all the way down to our manufacturing. And that's what we're here talking about today is our process development and manufacturing teams. We've got a wide pipeline feeding everything from early to late phase. Last August, we announced very positive news in our flagship product, which is SER-109. We saw very high efficacy. I think it was something like 88% efficacy in the prevention of multiply recurrent C. diff from going on further. This was a long road. We've been working on SER-109 since the start of the company back in 2012. And so it's like any new therapy. It's been a little bit of a roller coaster ride, and we were happy as clams to be able to have those phase 3 results. We also announced just today, so this is actually a really big day. We announced today that we fully enrolled our phase 2b study on SER-287, which represents our second place product. And that's for ulcerative colitis. And so the closure of that trial for further enrollment is a big step forward. Our phase 1 a couple of years ago saw a really great preliminary efficacy signal, and I think more important for patients, a great safety signal for patients. So if you know anything about ulcerative colitis, there's some aspect of the standard of care here which makes people progress to biologics which cause immune suppression activities, so that it limits the amount of inflammation in the gut. And those types of medicines tend to have some ugly side effects over time. And so if we can repair the microbiome and kind of fix the cause of the disease rather than treating the symptom, this will be a huge step forward for patients with UC. And then we've got a bunch of emerging programs. We started SER-301 trial as our first international trial, if you don't count Canada as a different country. So kind of ex-North America last fall, also in ulcerative colitis. And then SER-155, which is another infectious disease related drug for folks who are undergoing hematopoietic stem cell therapies. And we're hoping to start that later this year. But so those represent our core programs, and then we're also undergoing some exploratory programs in immuno-oncology. We think that there's a lot of research out there that's supportive that the microbiome actually affects the way that your body responds to a class of cancer therapies known as immune checkpoint inhibitors. You may be familiar with them. The pioneers in the field received the Nobel Prize for ICIs just last year, and so we're looking to add on to some of that research with MD Anderson Cancer Center to really investigate if modulation of a person's microbiome can actually affect how they respond to cancer therapies, which just seems like a very awesome possibility for those patients. So, I think why we're so happy with SER-109 as a flagship product is it really presents a proof of concept that we're able to modulate the microbiome and provide lasting lasting response. The changes that we see in the microbiome, even though our doses are only given over the course of a few days early in your treatment, a treatment regimen, we see that the lasting effect of that microbiome can go on for many weeks and months, out to, I think, 20 or 24 weeks has been recently reported. And so, having a new kind of therapy, a therapy that's been recognized as a breakthrough therapy by the FDA, that we can help these patients. And what that means is that that's a therapy that can be disruptive to the way doctors are able to treat patients. Specifically for this orphan therapy, where you've got 20,000 people dying per year in the U.S. from multiply recurrent C. diff, about 100 to 150 infections per year, and we're using that to provide proof of concept to the whole modality in general, and combine it with the safety profile that we're seeing, which is exactly what we would expect to see by replacing replacing a dysbiotic microbiome with a normal commensal wild type bacteria that were meant to be there in the first place. So it's a great combination for us that we're super excited about. And there was a paper, this paper that's listed here down at the bottom, Barb McGovern, who's our Med Affairs lead. We published this paper in April of 2020 in Clinical Infectious Disease. So if you'd like to see more about SER-109 and some of the work we're doing, including some of the scientific rationale, I encourage you to look up that paper. I did mention we're now a worldwide clinical phase company. Hopefully soon we'll be a worldwide commercial phase company. We work extensively with the FDA and Health Canada on North American based trials. Most of our trials have been North American based. And then recently we've expanded to Australia and New Zealand to work with the Therapeutics Good Association and MedScot in New Zealand, so TGA in Australia, to start that 301 trial, which is great for us as an expansion, being able to deliver these goods made in Cambridge, now delivered to patients in Australia and New Zealand, is a great step forward for what we're trying to achieve to make these drugs accessible to everyone worldwide. So we kind of picked the furthest place on the map and chose to go there. All right, so that's an introduction to Seres and some of our scientific work. I think Lisa's going to take it from here and talk about some of the core values and HR related items.
Thank you, Chris. I had actually asked Chris if I could lead with this slide because it very much resonates with me as a fairly new employee. And what I would say is when I was interviewing with a number of companies, and what really I think helped with the decision-making process for me around joining Seres was I saw these values exhibited throughout the interview process, and I was, and it was consistent. I met with probably, oh gosh, maybe 9 or 10 people, and we certainly wouldn't expect any of the students to have to do the same if you're interested in joining our company. But what really resonated with me was just this. I mean, people came across as very transparent, incredibly passionate about the science, and perseverance really came through for me as well. So there were quite a few folks that I interviewed with that had been with the company for 5 or 6 years. And so as Chris mentioned, I think he used the word roller coaster. So the biotech space can be a bit of a roller coaster, but the reward can be incredibly significant. And so I met with so many folks that had really persevered, and to see Seres 109, see the Seres 109 data and its promise, I'm really proud to be part of this team. So we work very collaboratively together. We challenge our people to think outside the box and make informed decisions, but also not be afraid of taking calculated risk. And I think that's really critical, frankly, in our industry. I think that's how we get to where we are today. And I also believe that, you know, we're a very transparent company, and I think you may hear this term quite a bit these days, but I really truly believe that what you see is what you get in people. And I also, I think, want to highlight that I believe that we have really brilliant folks here, really brilliant scientists and really brilliant folks across the organization. And there's a great sense of humility, and I really appreciate that. I'm not a scientist, I'm a liberal arts major. And so, you know, I spend time with the folks that are doing this amazing work, and they're so incredibly approachable and very interested in sharing their story and how they got to where they are today. And so we not only recruit against these values, so as we think about bringing folks on, we frankly look for people that embody what we believe would help them succeed here at the company, and that's exhibiting these values. So we're located at 200 Sydney Street. This is where our corporate headquarters are. And I actually have never set foot in the building because I assimilated during COVID. But we occupy several floors at 200 Sydney Street. And as you can see here, we're the anchor tenant, and Chris has outlined the various groups that are at this location. We are expanding fairly significantly this year because we're doubling in size. So today we employ 171 people, and we're expecting to double in size by the end of this year, and certainly do the same next year. So we're marching towards filing the company's first biologics license, where we're expecting to do that by the end of the year. And then, you know, hopefully the approval will be fast-tracked by the FDA, and we could potentially launch our first therapy in late 2022 into 2023. So it's a really, frankly, exciting time to be at the company, because we're not in our infancy stage anymore. We're not a fully integrated commercial biotech where we're in the adolescent stage where I say all the fun happens. So that's just a highlight of 200 Sydney Street, but we are looking at additional space. So we're outgrowing this facility. So we're looking at additional space for our R&D folks. And I think we have settled on his space, but we haven't made any formal announcement. And then we're also opening a donor facility in Cambridge and in Arizona later this year. So lots of growth, which presents tremendous opportunities for not only our current folks, but certainly new employees onboarding with us.
Yeah, and so as Lisa said, at this facility, we really built this to be a one-stop shop, which is quite unique in Cambridge and in the biotech community. Everything from initial discovery and research and development efforts through the technical development groups that take products that are handed off and are good. We start making prototypes of them, all the way through to our manufacturing and quality control facility are actually at this building as well. And so it's great because you get to see like a picture here on the left that you could walk across the hall and maybe see and participate in tech transfers and processes that are going over to manufacturing facilities or quality control labs that are just feet away, which is a great way for us to ensure that we're staying interconnected and understanding the challenges of both sides of the hallway.
And I think that's a really good point. That's a really good point to raise, Chris, because I think for folks, that presents a significant learning opportunity, because you don't always see these groups housed in the same location. And so that connection, to your point, is really a learning opportunity for people. It presents opportunities for sure. I think I might have interrupted someone. Did someone have a question? No. So this is just meant to highlight really high level our benefits programs here. I would say our total rewards philosophy at Seres is very competitive, considering we're a pre-commercial company, and there's very much a focus on the health and wellness and well-being of our employees. So again, this is just meant to highlight some of our programs. We recently launched an employee stock purchase plan where our folks have the opportunity to purchase Seres stock at a 15% discount. We do have a tuition reimbursement program here, which is frankly quite unusual for a company our size and within our life cycle. You usually see that much closer to commercialization. We have a pretty robust vacation program that includes a shutdown between Christmas and New Year's. We have we do have a 401k match, which again, I think is quite competitive given where we are as a company. And I haven't had the chance to participate in any of the employee activities, but I understand that we have quite a good time on site, and I expect we'll do so post-COVID as well. I heard the Christmas party or the holiday party is quite the bash, so I'm not sure that we'll get to that place again, but maybe some sense of the new normal. Yeah, and I thought this was really important to highlight. So, we are very much focused on diversity and inclusion, and it has really become front and center for the organization. We recently, we had a new head of HR join shortly before I arrived, and she has great passion and experience in this area. So, recently we had a number of activities to recognize Black History Month, and I'll just tell you about my own personal experience going through the month. I mean, there was a great deal of education that took place during the month, which I thought was incredibly beneficial for the organization, and we had great participation across the enterprise. So it's something that is front and center, particularly as the head of talent acquisition. I mean, my remit is certainly further expanding our sourcing and recruiting strategies to include a more diverse and inclusive candidate slate. We also do participate in Life Science Cares, which is an organization that was actually founded by the former president of Cubis Pharmaceuticals, and supports a number of communities that frankly don't have the same opportunities as communities that may be wealthier in nature or may have better school systems. And then, of course, we participate in programs and sponsor programs for the C. diff Foundation, the Peggy Lillis Foundation, and the Crohn's and Colitis Foundation.
I put myself on mute, sorry.
Okay.
Life at home. When I started talking, no one could hear me. So thanks, Lisa, for that. And yeah, I'll point out, we're very committed to Life Science Cares. Two of our executive committee members, Tom DeRozier, who obviously has worked with, well, not obviously to everyone here, but worked with Rob Perez, who's the founder of Life Science Cares, as Lisa mentioned, has worked with him and previously, but before Rob had started up this foundation, as well as Eric Schaff, our CEO, are both on the board of advisors for that group. And so that means we get a lot of encouragement to participate in their activities. Specifically here today, we've got folks from a subset of Seres, right? We call ourselves CMC, Chemistry, Manufacturing, and Controls. It's a little bit of an archaic term dating back to when most drugs were small molecules. But really, you know, how are we defining the chemistry of our drugs? Not just the prototypes that are being used to make proof of concept experiments, but really the chemistry of the drugs that are going into patients, and in our case, also the biology of drugs that are going into patients, and then combining that with manufacturing techniques and really controls to ensure that the patients are getting a safe and reliable version of that drug. You know, we've focused our history on clinical manufacturing. We've advanced one program now through phase 3 and now into a pre-commercial state. We've got other programs that are in phase 2 and phase 1 that we talked about. And we're looking to bring that out to emerging clinical or commercial manufacturing as that first program crosses that last hurdle into licensure. You know what? We end up being unique because we're dealing with a mixed live biotherapeutic product. It's some of the most complicated products that are on the market today. When you think about trying to make a pill that can stably deliver one storage over time, as well as stably deliver perhaps 20 different strains to the gut without significant loss of potency. And do so reliably for a wide variety of patients who may have different comorbidities or ability to take the drug. You know, we're working with hundreds of different bacteria. I think in the bioprocess group, Andrew has personally worked with 150 strains, which is some preposterous number of distinct. Sorry, not just strains, it's like 250 strains, but 150 or so distinct species, some of which don't have names because we're inventing new science, and when trying to import our drug to other countries, they ask us what the name is, and we don't have one for them, and so they've got to do their importation risk assessments based on sequence analysis. And that's a remarkable place to be. There's no literature, there's no playbook that we can go to for a lot of these things. And yet we're trying to turn that into a reliable manufacturing process to make the best drugs in the world for patients. And we
actually,
sorry,
just one comment I would make, Chris. You mentioned manufacturing in Cambridge, but we're also, just for the folks that are participating here, we also expect to identify another manufacturing site in probably the next 12 to 18 months, certainly as we look forward to commercialization. So that's certainly exciting as well.
Yeah, that's a great point. The facility that we've got in Cambridge, and then another facility that's a contract manufacturer of ours over in Europe, we're treating them as our launch facilities. And so not only are we looking to expand our R&D teams and our commercial teams and med affairs, which will help with patient and doctor education, but also our technology development labs and our manufacturing capacity, right? That every time we bring a new drug to the clinic or to the market, it's more manufacturing capacity that's needed, because we always need to manufacture our old drugs too. And so this presents a big challenge. The number of ideas that folks like Andrew can come up with far outpass our ability to manufacture them. So we've got to figure out how to make those reliable processes that are fired off correctly the first time, every time. And so it represents a really big challenge in the industry. And we don't have the luxury of having say for a protein, a monoclonal antibody expression, a platform process where we're expressing proteins using the same background cell line time after time, and kind of gives us a head start on the next drug. Everyone's unique, and we have to start over from scratch every time. And it's a big challenge. It's an exciting challenge, though. I like it. And so we highlight some of those challenges here. I think the appreciation of the complication of this drug is one that requires a little bit of historical context. There's a vaccine for pneumococcal meningitis that Merck came up with in the 90s, which was groundbreaking at the time, which requires 23 different fermentations to occur. And then they isolate the polysaccharides or some other antigen from those fermentations and combine them into a vaccine. You know, we're doing something similar now, except instead of just isolating a polysaccharide and stabilizing it, we have to stabilize the whole live cell because we want that cell to adapt over time. So if we're delivering dead cells, you know, we might as well be delivering placebos. And so this added hurdle of moving not just from a biochemical component of those fermentations, but to the whole live cell, just introduces the complexity on how we have to manage our fermentations all the way down to formulation and drug delivery. Also, we're dealing with spore-forming bacteria. For those who have maybe taken an internship or a co-op in industry, those are the bugs you're trying to keep out of your facility, right? Those are the bugs that can cause contaminations. They're the bugs that when you walk up to a contract manufacturer and you say, "I'd like to take a spore former into your facility," they say, "Your money's not good enough." So part of the issue that we've had is building our own capacity up, and that's why we really think we're special in terms of having capacity, you know, at all, let alone right across the street from all of our technology development labs. And we're doing all this, you know, with a high quality mindset. There's an article from 2002, but it's been repeated several times, where they tested the accuracy and precision, as you were, of probiotics. And what they found was that out of these European probiotics that were tested, perhaps half of them were mislabeled for what species they contained. So they said, you know, Bifidobacterium longum, and it wasn't, or they were containing missing or contaminating organisms, right? And so some other environmental organism had slipped past the goalie, or they just blatantly blended the wrong things into the capsule. And a third of them contained almost no live bacteria, right? And this is all the probiotics are all made according to you know, vitamins and dietary supplement standards. And so bringing up this level of GMP compliance that the FDA expects with a therapeutic good just raises the bar for us, but really ensures that the patients are going to get the drug that they deserve. I spoke about our quality mindset. You know, we have another set of core values that we think are quality behaviors that are key to a healthy culture. And what we're trying to do here is, and Lisa mentioned it briefly, was transparency. You know, we think that a transparent organization delivers high quality drugs to patients, right? And that's what we're trying to do here. And so we really empower everyone to speak up. You know, if in the subways, if you see something, say something. Same thing goes in our site at Cambridge, right? That we want to make sure that we're making drugs by the right procedures, and we're delivering the right drugs to patients first time, every time. We do this by educating our employees on what we expect and what the industry expects for our kinds of goods. We want to engage with our employees on a regular basis to seek feedback, to see if anything is what goes well and what goes poorly, and then motivate folks to really speak up, and that we recognize the people who are perhaps blowing the whistle on a you know, procedure or a process or a behavior which isn't meeting our expectations. Lisa, did you want to cover the COVID policies?
Sure. I mean, I think certainly what I want to emphasize is this is front and center for us as an organization. And I talked briefly about our benefits program, and of course the focus on the well-being of our employees, both the physical well-being as well as mental well-being. And so we have a COVID team that we put in place, and I'm not going to go through each one of these bullet points, but we have 2 weekly COVID teams that meet, and this includes a colleague of mine from HR as well as medical staff. So we have one team that supports really the on-site working interests of the lab, manufacturing, and other support staff. And then we have another team that evaluates the more, macro conditions. So what are the latest CDC guidelines, the relevant medical publications? We are constantly in communication with our peer groups to understand the protocols and policies that we have in place to ensure that we're implementing the latest and greatest. We're weekly testing all on-site personnel. The cleaning protocols have been really outstanding with respects to daily air purging and work area ventilation. And so I think what I want to emphasize here is that the focus has been on employee safety first and foremost. So the health and welfare of our employees is first and foremost, and we continue to ensure that we have the appropriate policies and programs in place to I guess, limit the anxiety amongst our employees. And so there's social and emotional support. We have an employee assistance program. Certainly employees have the opportunity to come to anyone in HR if they have some concerns. And then we're always open to feedback. So we've implemented a direct line that employees can reach out to, and so lots of efforts have gone into this, and we continue to monitor our policies, frankly, on a daily basis. As I said, it's really front and center and a priority for the organization.
Thanks, Lisa. And I think also in that interest of transparency, what we try to do is make all that information that we're uncovering in real time available to staff, and we do it on an internal Seres intranet page. Very early on in the pandemic, back in February, we had to conduct an on-site risk assessment for the pandemic because we are a manufacturer of record for FDA registered goods, right? For the clinical trial goods that we make. And I think what we were very happy to see is that when the FDA finally got around to issuing guidance in June of last year, we went back and back checked all of the things that we had implemented, and we were as good as or easily surpassed anything the FDA was recommending at the time, and we did so 3 months before the FDA ever put out guidance, and so that kind of speaks to our ambition to do things high quality and quickly, so that we can ensure that we are getting our drugs to patients on time. So we've mentioned now a couple of times we're rapidly expanding. We're super happy to be here and engage with the Amherst crowd. And what I'd like to do is invite some of them hiring managers that we've got on the call today to briefly speak to the types of positions you can find. I think there were several that were recently uploaded to your career services posting board just today. I think you'll find 6 of them up there. We also have additional job descriptions. We focused mostly on the entry-level positions, either for folks graduating with a BS or an MS, or whatever Amherst calls engineering degrees, whether it's BEs or BEngs or whatever.
Yes, BS.
BSs, they're all BSs, great. And so everyone's got their own little jargon in engineering departments. They're fussy crowds, those engineers. I'm allowed to say that. But so then you'll find some additional job descriptions on our website, and certainly, depending on how things shake out, we're going to be continuing to open new positions throughout the year. So if there wasn't one that you thought was interesting today, always please stay engaged with us, and maybe something will come up later this year.
Yes, and I would just if I could, Chris, I mean, I would encourage you folks to follow us on LinkedIn, right? Because you can not only, I encourage you to certainly check out our careers page on our website at serestherapeutics.com, because we're actively recruiting against 88 positions out there, or it might be 90 at my last count. So, but I also encourage you to follow us on LinkedIn and certainly on Twitter and Facebook to get all the recent news about what's happening at our organization.
Yeah, it's a great point. We also share articles and presentations as they come out. So we've recently presented at the Keystone Conference. We published an article in gastroenterology, including having the cover article in that journal, I think in early January of this year, and that was on our SEER 287 phase 1B findings. And so it's a great way to get information about us. We share directly through those platforms. Ariel, do you want to talk for a moment about the positions in analytical development and operations?
Sure. Yeah, so we're currently recruiting for an analytical operations associate or an RA position within analytical operations. So this role would largely be responsible for learning the novel suite of assays that have been developed by my group. It would also involve executing the assays routinely to generate, accurate, precise data, analyze the data. It's also very important to us to point out any trends that might be occurring, to really critically look at the data. So having that extra mindset for analysis, promoting any process improvements, authoring and updating SOPs and protocols. And just an overview of the kind of assays that we frequently work with in my team. They're largely microbiological based, given the nature of our products. It also includes things like CFU assays, microscopy, microbial ID, molecular assays such as PCR, biochemical assays, mass spectrometry. So, there'd be, of course, a training period associated with this role. Again, a lot of these assays are novel and specific to Seres, which is very exciting, that we've had the opportunity to kind of develop assays from the ground up. So that's pretty much what the role would entail. And if anyone has any questions, I'm glad to take them.
And this position is up in Handshake in our job board, so students can see the full description right there.
Fantastic. Let's give Andrew a chance to just hit the high points on the two bioprocess roles that I think got uploaded today.
They did.
And it's Amherst, Chris. Quick edit.
What did I said? Didn't I say Amherst?
Yeah, you did. Amherst. Sorry.
Where the H is silent.
Exactly.
Oh, yeah, but I'm from Philadelphia. We say water, so it's
Fair.
it's you gotta roll with the punches. I haven't assimilated to Eastern Massachusetts.
I'll get there.
Yeah. So we have a couple associate positions in bioprocess. Bioprocess is the group that sort of bridges R&D and GMP manufacturing. We take all these great ideas and we develop them into feasible processes. We optimize, we maintain inventories of research banks, research materials. We collaborate with formulations to, find ways to encapsulate these products and deliver them to people effectively. So yeah, I mean, like Chris said, we started our SEER 301 trial at the end of last year, and we're hoping to start the SEER 155 trial soon. So our group is actually already developing phase 2 processes on many, many different bacteria. So there's a lot of work to do. We need help optimizing things. We need help automating things, purifying things, controlling the processes, all of the above.
Yeah, there's no shortage of work, especially with doing all of this times 50 strains or whatever we've got to do.
Exactly.
Thanks, Andrew. Melissa, we've got two, I think, one for donor operations and one for donor sciences. Would you like to speak to your group and those positions at a high level?
Sure, yeah, and actually both of those positions are donor analytical sciences, junior level coming in. So my team, it will be growing. I'm building out kind of a new team. And our role is really screening microbiomes of potential donors that basically to feed our pipelines. So interesting work. A lot of microbiology. We'll be expanding into some other assays this next year as well. So there's going to be some assay development opportunities towards higher throughput. And then also inventory management and interacting across the company. I think that these positions as well, pretty much all of these positions at a company that's growing like we are, is a great way to start your career when a company where you're learning the industry, learning about industry, and being at a company where it's growing and you have the opportunity to learn what other groups are doing, as Chris was kind of describing about going across the hall. But really a great way to figure out what you really want to do. Get your foot in the door, figure out what you want to do, and move around the company as you find your fit. So I'm going to be hiring probably about 6 people this year, entry level. So keep an eye out and hope to see some resumes. And if you have any questions, I'm all over it.
Thanks, Melissa. We also have a last position that's up on Handshake right now, which is this R&D position in our screening and microbiology group. Didn't get them on the call today, but basically this is the group that feeds Andrew's work in bioprocessing. They're working with our discovery teams to isolate new and novel kinds of bacteria, quickly characterize them as best as we can, but then launch them with great speed towards Andrew's media development pipelines and bioprocess development activities. So you'll find that one up there as well. Great. I think we've got some time. I don't know if any questions have come in or if folks have anything coming to mind now.
Yeah, Chris, we got a question. What's the difference between the research associate and the engineering associate position? Do you want to answer that?
Sure, we'll take that one on. I think the main difference here is a focus on skill set and education. The bioprocess team is a mixture of folks from the sciences as well as engineering. And so, folks on the research associate side, we would expect to have more of a background in biology or analytical chemistry or some other physical science. Whereas the engineering associate, we're going to ask to do work with bioreactors or pumps and filtration that are probably going to better align with the education they're going to get versus the microbiology education that we need on some of those other activities. And so that's the differentiation. In practice, we work side by side. I think we put Andrew on a kinetics project for his first tour in the laboratory, which was mean and cruel. And he said that maybe he took calculus once, but we made him prove it. And so, so in reality, there's a lot of cross training that goes on, and it's really not an exclusive difference between the two. It just helps us get to the right hiring managers more quickly.
I have a question, if that's okay.
Yeah, absolutely.
So it was mentioned multiple times that you guys are expanding overseas. I was wondering if there are currently any entry-level positions on either the biology side or the business side overseas.
Yeah, great question. So a lot of on the business side, a lot of our work now with SEER 109 is handled with a partnership with Nestle Health Sciences. They were a very early funder of ours. They participated in our Series C before we went public back in late 2014, 2015. And so the rest of world distribution will happen according to that Nestle partnership for these early drugs. What we've got is some engineering support positions, and you'll see those, I think, in the emerge in the manufacturing team over the next couple of months. I don't think there are any positions open now, as we need to work with our contract manufacturer that's over in Europe. And so those are likely positions where it'll be extensive travel to Europe to the site. It won't be probably a permanent relocation because of the difficulties around work visas and other things. And so we tend to keep it towards Cambridge being folks' home base, with really like extensive travel, several weeks a month.
Okay, thank you.
Yeah.
Are there other questions from those who are still able to be here? I have some questions that I think students might be interested in knowing. Will you hire international students who have OPT, that optional practical training that comes with their student visas? And as a STEM student, they have 3 years of US work authorization before they need to be sponsored.
Yeah, so if folks have come with work authorization already, we have supported them in the past.
Okay.
The new work authorization is something that we don't have a great system for through our legal team to be able to support.
Questions?
I have a quick question, if possible.
Please.
I just didn't get quite an understanding of what the title or job role description would be for someone who's traveling a lot for you guys. Could you just quickly say that?
Yeah, so it's it'll be under the when it comes up, and I don't think there are any listed right now. We would have probably posted that. It would be under our MSAT group, which is called Manufacturing Sciences and Technology, and you'd look for like an Engineer 1 position that mentions our contract manufacturing facility and support of our CMO, not Chief Medical Officer, but contract manufacturing outfit. And so organization. And so that's what you would be looking for there.
Thank you.
Yep.
So Julia has put in an email in the chat box for those who have attended this. They could send their resumes directly there, correct? But that means they still need to apply directly to a position as well. Correct.
Yeah, I think that's what we would be prefer. If for some reason, we do keep a repository of maybe things that are mismatched. And so if you don't see something that you feel match, although these are fairly entry-level positions that we would expect to be suitable for most folks in freshly graduating, matriculating from school. But yeah, if you'd like us to have your resume on file, it'll be most efficient to probably go through the application portals. But if you'd like us to have it on file for other purposes, please feel free to send it along.
Anyone else have any questions? So I want to thank all of you for attending, both from Series and our students. And I know I'm watching my email of people saying, I couldn't attend. Can you send me this? Send me that. I will surely highlight these positions in our weekly newsletter, get it out to students. You have great opportunities for our students. So there's a lot we can continue to do together. I'm happy to stay on afterwards. In fact, I'm going to turn off the recording